Welcome to solverfy.
So what I'm drawing here is a single bacterial cell. And what we're going to do is we're going to expose this bacterial cell to acid, which is simply an excess of hydrogen ions, drawn here as H+. Now, the hydrogen ion concentration, or pH, within the E. coli cell is relatively constant at about 7.5. But in response to an acid treatment, some of these hydrogen ions are able to diffuse into the cell and increase the hydrogen concentration, which would result in a drop in pH. Now, what's interesting is that the cell is able to recover very quickly and return the pH back to that critical value of about 7.5, which means that it must somehow be controlling the level of H plus ions within the cell. So the question also mentions that ATPase inhibitors block the recovery of this pH control. So to understand that, we need to know about ATPase enzymes. So these are membrane-anchored enzymes, which in our cells they're present in the inner membrane of the mitochondria. And what they do in our cells, in a mitochondria over here, for example, is they allow numerous H plus ions to flow down their concentration gradient and be released into this lower or higher pH environment. But in the process, they will form ATP. So in the example of the bacterial cell, it's possible that these ATPase enzymes are acting in one of two ways, in two potential ways. So one would be that they harvest energy when they're exposed to a high pH environment. So if they're shocked with acid and the ATPase were in this conformation, it would enable H plus ions to flow into the cell, but it would enable the formation of ATP. And this ATP could fuel a hydrogen ion exporter to carry these H plus ions out of the cell and reduce the pH. Alternatively, the hydrogen ions could enter the cell passively by diffusion, and an ATPase enzyme in an inverse orientation could actually be the enzyme that pumps hydrogen ions out of the cell. But notice in this case, it would likely be against their concentration gradient. So the key take home from this question is that to maintain a constant intracellular pH, there must be an active mechanism for controlling H plus ions. If it were just done passively, just done by diffusion, then the pH would just match that of the environment. So since the pH is constant, irrespective of the environment, it means that the cell is somehow pumping out H plus ions. And there are two options. Either the cell is using ATPase uh, enzymes to harvest energy when hydrogen ions flow into the cell and using that energy to pump hydrogen ions back out, or it is providing energy to an ATPase molecule itself, which is pumping the ions out.